Hello. Now we are going to see the eukaryotic cells, right? In eukaryotic, how this process of transcription takes place. Okay. In eukaryotic, uh, the nuclear polymerase in eukaryotic is three types. Polymerase 1, polymerase 2, RNA. RNA polymerase 1, 2 and 3. But whereas in prokaryotic, that is in bacteria, we have seen RNA polymerase is only one type which does all the work. But here, there is a division of labor. That's why they are higher organisms. Okay? So, they does the work beautifully. This RNA polymerase 1 prepares tRNA. Sorry, it prepares rRNA. rRNA, there was a doubt. And this prepares the mRNA. And polymerase 3 prepares tRNA. See, beautifully they have made some differences in the enzymes, and RNA polymerase a separate enzyme which involved in the process of rRNA synthesis, and it has a smaller and bigger residues, and it produce not only rRNA along with its particles, right, 20S and 80S, 28S, it is not 28, and here it produces 5S and smaller nuclear RNA. This is produced here, and RNA polymerase 2 produces mRNA. So this mRNA actually, before it is producing, actually it produces a precursor that is called HNRNA. What is HN? Heterogeneous nuclear RNA. It's a precursor. All this process takes place in the nucleus. When it comes out, it has some changes. So now there are three types of RNA polymerase. RNA polymerase 1, which actually transcripts, don't say prepare, transcripts, the same word can be used here. They transcript rRNA, they transcript mRNA, they transcript tRNA. So this is a small introduction. Now we are going to see how this process of transcription takes place in eukaryotic, right? Right. The process of transcription in eukaryotic. Prokaryotic we have already seen. Now we are going to see eukaryotic. Before that, I just want to tell you, as it is a higher animal, I just explain. This is nucleus, where DNA is present, and all the protein synthesis, mRNA, rRNA, They are all here. All these RNAs are here, which is going to do protein synthesis. To prepare this mRNA, tRNA and rRNA, the DNA does the work inside the nucleus. For that, it has three enzymes: RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. Right? Clear. Now, the DNA cannot come out, as I already said in the transcription beginning stages, and it sends the mRNA first out. And this is what the process which we are seeing it as transcription. And as it is a higher animal, the DNA, it is actually a DNA only replacing the thymine. Wherever thymine is there, uracil is replaced and it becomes mRNA, right? And I will tell you, suppose you think this is a king and this is its kingdom. The king wants to come out, everybody can identify this is king. So what the king does, when the king wants to go out and just check what is happening in the kingdom, the king does a different makeup, change his complete features and comes out where nobody can identify this is the king. Just think this in your mind. Now I am telling, DNA is the king. It wants to come out and give the instruction and make it does some work. It cannot. So it makes some changes. That first change is the king, how it does, he makes some artificial moustache, beard, something like that. So here the king first replaces thymine into uracil. That's the first step. Okay, the makeup starts. Okay, now I am going to teach you how the DNA made its makeup. You think in this way, it's very easy and simple. But if you see it very scientific, you may find it difficult. Now I am going to teach you how the king comes out as mRNA in the name of mRNA. Okay? With some makeup. There are 4 steps in the makeup. Right. The first step in the makeup, step 1. Suppose the king, you just imagine king made some haircut and make his hair something different. Okay? We call it as splicing. It is nothing but trimming. Some trimming work takes place in the DNA. How this trimming takes place and how this, what is actually formed in the nucleus is, I said already, HNRNA. That is heterogeneous RNA. Heterogeneous RNA is nothing but precursor. Precursor is which is going to be mRNA. Which is going to be mRNA. In this, there are 4 steps. 1, 2, 3, 4 steps which we are going to see. That is makeup. The makeup 1 is splicing. What is splicing is, if you see this HNRNA, I'll draw HNRNA. I'll draw it like this. HNRNA. At places, it has codes. Okay. Some places it doesn't have codes. This is how the HNRNA is there. This is HNRNA. Okay. HNRNA. And what is this two different? The thick one and the thin one. The thick one is with codes. Some codes are there. And these regions, there is no codes. These regions are with no codes and these regions are with codes. And in the HNRNA, this is HNRNA. This HNRNA with the coded region are called exons. Okay. The H in RNA without codes are called introns. Now there are two names, introns and extrons. And this is an unwanted region. Okay. So we have to remove this. That is what is trimming, cutting. Cut this, join this, cut this, join this, cut this, join this. This is the work takes place. That is called splicing. I said it is trimming. Okay, unwanted are removed. That's the second stage. When unwanted are removed, what happened? Every time introns are removed and exons connected. This process is called slicing. First slicing takes place. When slicing is over, how it looks? It looks like this. So there is no this gaps, this white, this is completely, now trimming is done. Some makeup work is over, slicing is over. Okay. And this, we call it in two names. I'll just tell you the names. Okay, now we move on to the first step is over. This is H and RNA. This is nothing but heterogeneous RNA. It's a precursor. Precursor is prior to mRNA, which is going to make mRNA. This is going to make mRNA with some changes. These changes, which I have started one with the name called splicing. Splicing is nothing but the H and RNA has two regions. Regions with code, regions without code. The regions with code is called exons and introns. And the introns are replaced, removed, and exons are connected. And this H and RNA now become like this. Now H and RNA is like this. Now half of the makeup is over. Now the next makeup. Now I want to send this beautiful H and RNA to out. Okay. So while you're going out, as I said, some dressings is to be done. And here, say for example, I'll give you an example. You put a cap, okay, to hide your hairs and all. Put shoes and all, okay, then you look different, right? So now these two steps takes place, which is also named in the same way. What are the next two steps I will show you? Second step, capping. Name is capping. Okay, capping takes place. Untailing. Untailing takes place. Capping and tailing. What is capping? Okay, capping is nothing but in the beginning, I am adding some factor. This factor is named don't get fear, this is very simple. It is methyl. Always methyl group is first. Methyl guanine triphosphate. Methyl guanine triphosphate. Triphosphate is written as PP. That means 3 phosphorus. That is what is written. When this methyl guanine triphosphate join at the beginning of the HN RNA, that is called the capping. Okay, now capping is done. The next is tailing. Tailing, lot of residues, the unwanted residues get joined here. Lot of residues joined here. And these residues are nothing but adenylate compounds, adenylate residues. Around 200 to 300 residues join, and it join at the end. This is called tailing end. Once this is done, now you see, previously the HN RNA was like this with the white color, orange color, white, orange. Now what happens? Splicing takes place. The extrons are removed, introns are connected, and after connecting, a cap was made, and at the end, the tailing was done. Now this is coming out of nucleus. This all happened in nucleus. Now, when all these are happened, now you cannot call it as HNRNA. Now we call it as mRNA. These are the steps involved. As I said, the king come out with the different makeup. You see, the king was in a different. Actually, DNA was different, and DNA makes HNRNA. The HNRNA makes some trimming work. Then it makes a cap and tail. Then it comes out as mRNA from nucleus to cytoplasm. Okay, this is the actual steps involved in the transcription. There are different steps involved. Okay, I think you understood. The king come out and ready for protein synthesis. It joined with tRNA and rRNA, prepare protein synthesis. Our next topic is